Hello everyone, and welcome to today's webinar, Advanced Phenotyping of the Tumor Immune Microenvironment. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by UltiView. To learn more, visit ultiview.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Angela Fasaturo, Associate Director, Biomarker Strategy and Applications, and Dr. Fabian Schneider, R&D and Product Management at Visiopharm. Dr. Vasaturo, you may now begin your presentation.
Thanks for the kind introduction and good afternoon, everyone. Today I'd like to start with a quick introduction on the tumor immune microenvironment. As you can see in this very elegant representation, the tumor microenvironment is extremely complex. It not only consists of tumor cells, but also many other different cell populations, including cells of the innate and adaptive immunity that play an important role, especially in response to therapy. And it's actually this complexity that drives the need of using multiplex immunohistochemistry that allows the analysis of multiple immune cell phenotypes and their spatial distribution and interactions. Multiplex IHC becomes also critical when working with very precious clinical samples, trying to provide data to make a decision on whether or not to move a drug forward. So in this context, the selection of the appropriate tissue biomarker coupled with the spatial analysis becomes really key for generating as compelling as possible data from the very few samples that are available at this initial stage of clinical trials. And this is not always the case. So data are sometimes inconclusive or the biomarkers that have been selected are not predictive, or simply the assay shows not to be reproducible, or is too complex to move in the path towards clinical development. So when working with patient material, you want to make most of the tissue you have. Therefore, you want to have the full slide analyzed. You want to be able to look at the spatial relationships in the tumor microenvironment. You want to generate highly consistent data. And ideally, you want to combine your biomarkers of choice with H&E on the same slide. And H&E on the same slide is extremely powerful when trying to scale to larger studies, but also when you want to simplify the assay based on promising indications that the drug works. So when you want to work with clinical trials and you want to optimize the chances of being successful, you need to start from the selection of the most powerful biomarkers by testing multiple competing hypotheses at the same time. This is why we like to have up to 8 biomarkers on the same slide plus an H&E on the same exact slide to be able to generate consistent data that are also critical for algorithm training. Multiplex IHC is often still perceived as a very complex technology, and although the TME is complex, your assay shouldn't be. And this is actually the aim of the in-situ-plex technology that UltiView has developed, that made really multiplex simple and allows to go from tissue to data in a one-day workflow. So the staining is pretty simple and takes very short time and allows for whole slide imaging and the very deep phenotyping and spatial mapping that you will hear about in the next presentation. Here, a very quick overview of the in Duplex technology that really follows just a few steps. The first one is an antigen retrieval, only one, that allows the preservation of the tissue integrity and the H&E on the same exact slide where we perform fluorescent multiplex IHC. Then a staining step where we incubate the sample with a mixture of DNA barcoded antibodies, followed by an amplification step where the DNA barcode is elongated to allow more complementary fluorescent DNA probes to bind in the last step, which is a detection step. And at this point, your samples are ready to be imaged. So with this simple technology, there is a complete retention of sample antigenicity, and the morphology of the tissue and the architecture of the tissue is well preserved. That allows H&E on the same slide, and therefore it's fully compatible with the conventional pathology. It is also a very high throughput technology that can be applied across multiple tumor indications without the need of re-optimization. And it can be also easily transferred to CRO outsourcing partners for clinical research implementation. So this slide summarizes the initial LTV offering of the in Duplex technology. Here you see different Ultima Per kits that are all 4-plex kits that include a different combination of markers that allow to explore complex phenotypes and allow to answer specific biological questions on the tumor microenvironment. For example, there is a kit that looks around antigen-presenting cells or myeloid derived suppressor cells, or also activated T cells, regulatory T cells, but also phenotypes along the PD-1 and PD-L1 axis. More recently, this standard offering with the off-the-shelf kits has been extended to an additional kit, which is now an 8-plex panel that we call Ultima Per Immuno 8, that I will focus on in the next couple of slides. But the LTV offering is also extended now to custom assay development. So we have a dedicated assay development team that can build your custom panel up to an 8-plex in 8 to 13 weeks in total. This is an overview of the Immuno 8 panel that we have recently developed that includes the main phenotypic and functional markers for the key immune phenotypes that are critical in the tumor microenvironment. What I would like to highlight here is that as for all the other kits and also for the custom kits, the panels that we develop can be applied to multiple indications without need of re-optimizing any conditions. And here you see an example of the Immuno 8 panel run on multiple tissue types, including non-small cell lung cancer, colorectal cancer, and breast cancer. And in the next couple of slides, I will focus on the application of the Immuno 8 panel on colorectal cancer tissue. So on this slide, you can see on the left side an overview of a colorectal cancer tissue stained with the Immuno 8 panel, and a zoom on one leader area where you can see some tumor nests, but also a lot of infiltrating cells, immune infiltrating cells. And now the Immuno 8 panel includes markers that allow to identify the key T cell phenotypes, including regulatory T cells, exhausted T cells, cytotoxic T cells, but also immune-evading tumor cells or immune-suppressive macrophages. So it really gives a lot of information on the dynamic nature of the tumor microenvironment. And again, based on the combination of different markers, including CD3, CD8, CD4, FOXP3, PD-L1, PD-1, CD68, and cytokeratin, you can test multiple hypotheses at the same time and identify really the meaningful phenotypes, the meaningful biomarkers that give clinical utility. And here I just wanted to show you a couple of examples of the quality of the staining, but also of the different phenotypes that you can identify. Like in this image, in this slide, you have CD3 in green, and then you have CD4 in red. So the combination of CD3 and CD4 allow you to identify T helper cells. And then if you add FOXP3 in white in the nucleus, then you will be able to look at Tregs. And in the right panel here, bottom right panel, I have added just the cytokeratin in cyan to be able to show where those different cells are located. And as you can see, there are some that are really intraepithelial, so intratumoral, and some others that are really between the different tumor nests. In this specific case, for example, I have highlighted the cytotoxic T cells identified by the combination of CD3 in green and CD8 in red, but I have also added on top of it the PD-1 in yellow that allows the identification of exhausted T cells. And in this case, you can see that most of them are CD8 PD-1 expressing cells. Again, the cytokeratin addition to be able to look at the localization of the different phenotypes, and as you can see, there are multiple PD-1 positive cells that are located within the tumor nests. Last, you see in pink here PD-L1 staining and its combination with either CD68 in white or pancytokeratin in cyan. And this allows you to identify really cells that express PD-L1, which are not necessarily only tumor cells, but also can be macrophages. And in this specific tumor, you might be able to see in the lower left panel that most of the PD-L1 is expressed on macrophages rather than on tumor cells. And in the bottom right panel, I have added PD-1 so that we can look at the interaction between PD-1 and PD-L1 expressing cells. And again, I have kept the cytokeratin staining to be able to look at the specific localization of those positive cells with respect to tumor. And with these slides, I really want to summarize and reiterate the concept of the power of using an 8-plex assay to test multiple hypotheses at the same time on the same exact tissue section, and then select the most promising biomarker subset that are the ones that would give the clinical utility. So eventually what you would do is testing the multiple hypotheses by using up to 8 biomarkers, but go down in the number of plex, in the number of biomarkers that are really meaningful to your trial, and eventually transfer this to your CRO choice and for clinical deployment on a larger cohort. So although we of course highlight the power of multiplex, we still believe that ideally you want to get to the least number of plex and the least number of phenotypes that are really able to give you the clinical utility that you look for, and that can be easily deployed in a larger study at the CRO. And last but not least, I have mentioned previously during the presentation that we can perform H&E on the same slide where we perform multiplex IF. And again, this is extremely powerful, and we can do that because of the type of assay we are using that preserves the tissue architecture and the cellular morphology. And this is really not adding a lot of time to your staining procedure, and it follows the exact same protocol that is used in pathology labs. But it is something extremely powerful, especially from the fact that it adds the phenotypic and functional information to an image that is what pathologists look at, and gives them the morphological features that are eventually driving diagnosis, for example. So if you look at this image, a pathologist would be really comfortable in saying what is tumor and what is stroma, but also it would be easy for a pathologist to say as well that there are a lot of infiltrating cells. What becomes complicated is saying what is the function of those cells and if those are exhausted T cells or if they are cytotoxic T cells. So, and this is where the additional IF data and information come in, and the combination of the two is really what gives what takes most out of a simple tissue that can be extremely precious. And with this, I'd like to thank you all for your attention.
Thank you very much, Angela, for the great introduction into the IO8plex and your technique. My name is Fabian Schneider, and I will show you how the VisuPharm platform can be utilized to decipher all the phenotypes that an IO8plex or an 8-marker multiplex assay delivers. So in the background, you already see the CRC case on which we will demo our capabilities of our platform. And you already see quite an overlay of markers and where the cell types are touching each other. In immuno-oncology, the cellular phenotypes of the immune system and markers on the tumor are of utmost importance to understand mechanism of actions of new drugs. And for this, we need to understand where are the cell types and their interactions. What are the distributions of these phenotypes that are resulting from the markers? Where is the receptor and ligands in the immune cancer synapse, and are they correlating? I would like to introduce you to the VisuPharm software platform, which is a toolbox for image analysis scientists, in which you can combine the rule-based machine learning with artificial intelligence deep learning classifiers. All your outputs that you would like to generate are fully customizable, and no coding is required from the user. And we have a lot of modules with which you can extend the functionalities of your platform. For example, tissue array, in which you can use your tissue microarrays in bright field and fluorescence. You can de-array the cores and for an individual analysis of each. We have the multiplex phenotyping module, which gives you an automated unbiased clustering tool for the analysis of high-plex data. We have a tissue align module with which you can co-register serially sectioned and stained images across modalities like IHC and H&E, IHC, H&E, and multiplex IF. We also have one product that is called the Brain Mapper for the mouse brain, on which we have a pre-built atlas with deep learning classifiers to identify the mouse brain regions, horizontal and sagittal sections, and you can identify then your biomarkers and quantify them within the different regions. Today's problem is the immuno8plex and the phenotype analysis. So the first thing that you need to do is you need to segment the tissue into the blue here, the cancer, versus the stromal regions. Then what you would do is you do a second app, is cell detection. So you need to identify all the nuclei, and then you can expand the nuclei into a cell segmentation, and then you can phenotype with the underlying presence of the biomarkers, which will then result into many different classes of positivity for these biomarkers. And we offer this based on machine learning classifiers, and we can combine this with AI deep learning capabilities. And then once you have your cell segmentation, you can run an automated phenotyping with the phenotyping module. What we have used for the tissue segmentation as well as for the cell segmentation, we have used the AI deep learning classifier. What you will get out is simple results like box and bar graphs that you can report the positivity of cells or percentages or densities. You can show the results also as heat map overlays on the tissue directly. to display the distribution of your phenotypes across the tissue. Another thing is that you can calculate all the distances between classes, so phenotypes or regions, and can export this into into your output files and then generate distance measurements. And further on, you can also display all your phenotypes in phenographs. So this is a dimensionality reduction, where you can look at the display and the clustering, and you can look at the fingerprint of your tissue samples and compare them to each other to see differences in the combination of the phenotypes within the tissue. The first thing you do for the image analysis is to limit your analysis to a region of interest. In green, we have detected automatically the tissue and have annotated in blue here on the IF image the region of interest for the tumor region. An expert pathologist can also do this as offered by UltiView directly on the H&E stained tissue image that is available together with the MIF on the same section. After annotating the region of interest, we use the cytokeratin channel to generate with a deep learning classifier a CK segmentation within the region of interest to separate the tumor epithelial region from the stroma. The invasive margin around the tumor epithelial segmentation is shown here with two margin zones of each 50 micrometer diameter, one in cyan and one in red, which will later be also used in the data analysis, where you can then show the distribution and the densities of the phenotypes within these different areas. If we look in more detail at the tissue segmentation and the invasive margins, you see in blue the CK segmentation. And also within these margins that are far from the tumor mass, you see some single cells that are CK expressing. These could be either invasive cancer cells or these could be reactive stromal cells. But the more you find of these, and you can quantify this then with our software, the more you can look into the invasiveness of such a cancer case and give a predictive, and this has a predictive value for the survival of the patients. The immunohistochemistry, as already shown by my UltiView colleague, is super high plex and very information dense. So if you look at all 8 markers at once, we are not cognitive capable to sub-identify and decipher this image. So when you look at this region and we only look at the lineage markers, then we already see that a lot of these CD4 expressing cells and are surrounding together with the macrophages the tumor islands here. So they are like delineating it. And you see within the tumor epithelium, you see CD8 positive cells. On what I want to focus today is the cytotoxic T cells, and when they are becoming either exhausted or primed with the expression of PD-1, which is available in this kit, and macrophages and tumor cells that are expressing PD-L1. So the first thing we are doing when we want to do the phenotyping, as said, is that we have to segment the cells. Here you see the result and the layer mask of the cell segmentation for the whole slide and for this region. Then we do the phenotyping on top of that, and here I'm only showing you some lineage markers. So you see here are our macrophages, here are the tumor cells in purple, we have the CD8s in green, and we have CD3 in cyan and CD4s in pink. And we can already see that macrophages seem to shield the tumor from the invading T cell compartment. If we add on top our functional markers to the lineage markers, we see that the FOXP3 positive cells are intermingled with the macrophages, and the PD-L1 positivity is mainly on the macrophage population in this case, really shielding and delineating exactly at the line of the tumor epithelium, the tumor from the T cells, and PD-1 expressing cells are either in contact in the invasive margin with these PD-L1 expressing macrophages, as you see, or are on the tumor. We can also visualize this a bit more easy by only showing with our software dots on top of these cells that you can then give specific colors to see the grouping of your cells. Here, as said, I would like to focus on the capabilities for distribution of these biomarkers. So you see the heat map for cytokeratin on the left. In the middle, you see the heat map for CD68 for macrophages and CD3, CD8 double positive cells. The heat maps are all normalized so that the yellow color is around 300 cells per square millimeter density. If we then add the functional marker PD-L1 and PD-1 on top, then you see that CK positive PD-L1 double expressing cells are mainly in the region down here, and some scattered cells in the tumor, while CD68 PD-L1 double positives have a hotspot also correlating with the CK expression and are around the tumor islands that are towards the larger stromal areas. And the CD3, CD8, PD-1 triple positive cells, these are mainly around the whole tumor tissue and are also on the tumor epithelium. We can also combine the two heat maps for visualization, and by multiplying PD-L1 here with CD8 PD-1 double positives, and this leads to this phenotype clustering where you see that the spatial correlation has the highest possibility around this area and around these two areas where you have the CK and PD-L1 expression and the PD-L1 high macrophage regions. When we are exploring the tumor epithelium versus the stromal densities of our phenotypes, we can clearly see that in the stroma we have higher expressions of the T cells compared to the tumor epithelial area, and we also have more macrophages. In both populations, CD68 only or double positives with PD-L1, so the more M2-like phenotypes in the stroma. For the CD8s, this is the opposite. So we have more CD3, CD8s that are unprimed within the stroma, and on the tumor epithelium, most of the CD8s become positive. We can also look at more rare phenotypes that are identified. For example, CD8 cells that express FOXP3 and CD8 FOXP3s that are also PD-1 positive, or CD4s and CD8 double positive cells and CD4, CD8, FOXP3 triple positive cells, etc. But if you think back into the marginal zone segmentation we did, we can also investigate where are these cells at the highest density. Is this on the tumor, as for CD8s with PD-1 expression? And there we see that we have a clear draw between the inner invasive margin and the and the tumor epithelium, while the CD3, CD8s are also more distributed across these three areas. This is also similar for the CD68s that are PD-L1 positive, so they are similar in density between the two margins. For CD8 and PD-L1, a direct interaction is important. Therefore, we can also do spatial analysis of our phenotypes by looking at the cell-cell contact. For this, we use our cell segmentation mask and then bring out the phenotypes, so only the CD8 expressing cells. And then we look at all cells that are in direct contact with these CD8s. And then we can take these as another class out and sub-phenotype these then further and look which are the cells that have the highest frequency in contact with our CD8s. And this we can do for all phenotypes, and we can do these experiments over and over for all of the resulting phenotypes, and then compare across cases. Visiopharm's platform offers multiple possibilities to explore the represented classes in the whole slide tissue or your biopsies or tissue microarrays. So you can look at phenotype profiles where you see the percent of positivity and that is identified of these different phenotypes as a bird's eye view bar graph, heat map, for example. We have the possibility to also use t-SNE dimensionality reduction to investigate the clustering of different phenotypes and also to look at the fingerprints of your samples. You can then also look at the phenotype matrices, which phenotypes are expressed within your cases, and you can also look at the neighborhood plots that is available within our software to understand if your cytotoxic T cells are clustering, for example, with proliferating cells in general. With this, I hope I could show you that the Visiopharm multiplexing analysis capabilities are powerful, and that with the IO8plex by UltiView, we can explore a lot of phenotypes and also identify them and look at the interactions of all of these cells. So I only showed you one aspect of looking at PD1, PD-L1 axis on cytotoxic T cells and macrophages and tumor cells, but you can look at all different combinations like FOXP3 CD4 distances to CD8, or the clustering of PD-L1 CD4 double positive regulatory cells or T helper cells to CD8 PD1 expressing cells. And what is this relation to the general PD-L1 expression? You can look at all different distances on which you are interested in and get them as an output. And you can compare all of this to other cases. So you can apply the same algorithms to the same assays on the same indication, or tweak for the next indication and move your analysis forward. So Visiopharm offers this flexible toolbox to build your own apps, so algorithms, and workflow to answer your specific scientific questions. You can use deep learning classifiers and train them by example, by defining regions for the analysis or measurements. All the app workflows are fully customizable and all outputs are accessible, and you get the numbers you need or export the entire data set. So you are not limited to that. And our platform is fully integrated with the multiplex phenotyping workflow for an automated phenotyping. With this, I would like to thank you for your attention and thank UltiView for giving us the opportunity to present our phenotyping capabilities for high-plex immunofluorescence assays. I would like to thank our product team for the phenotyping in computer vision and AI at the Visio and David Mason for providing me the skeleton for this slide deck. Looking forward to the Q&A session together with Angela, and looking forward to your questions. Thank you.
Thank you, Dr. Vasaturo and Dr. Schneider, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Okay, our first question is, have you tried also other stains besides H&E after the UltraView procedure, such as the good quality PCR?
I guess that's for me.
SR, I'm sorry.
Yes. So we haven't tried it, but I don't think there would be any issue because basically we have used H&E since it is the most commonly used staining in routine pathology. And basically proves the fact that we are not affecting the tissue in any ways in terms of architecture and cellular morphology. So I guess whatever other staining like the one that was mentioned here, I guess is the Picker series red for collagen fibers should be something applicable, but we haven't tried it.
Thank you, Dr. Vasaturo. The next question is, how does the antigen retrieval work?
One more question for me. So it's basically the classical antigen retrieval that you would perform also for chromogenic IHC, like DAB staining. So the protocol that we use is fully optimized to run on the BondRx. So usually this is here 2, which means pH 9. But it's really, if you would do it manually, any of the classical antigen retrieval can be used.
Okay, thanks again, Dr. Vasaturo. Let's see. I think that Dr. Schneider is still trying to get his audio connected for the Q&A. So let's see, if I ask a question that's for him, will just let me know and we can postpone that until he's connected. So our next question is, would this system be cost-effective for large sample sets, i.e. 70 samples, or better suited for smaller amounts of slides?
I'm not sure if this is a question related to the staining portion or the analysis portion. And I think it's a question that might be, if it's staining related, it might be a better question to be addressed afterwards with the sales team. I can't really comment on costs.
Okay, thank you. We really appreciate that. The next question is it the Phenographs module on market? Can you explain a bit more what it is? I was just wondering if this is the clustering algorithm or something else.
Yes, so now I'm back. So my internet connectivity here in my holidays site is very bad. I'm sorry for that. Yes, the Phenograph model is on market, and this is an automated clustering with using a Gaussian mixed model. And we are currently working on updating that in the future.
Oh, great. We're glad you're back. Thank you, Dr. Schneider. Our next question is it possible to visualize and analyze of any particular biomarker combination on aggregate data from different groups of tissues, e.g. tumors from treated and untreated tumors?
Yes, if the assay is the same and the indication is the same, it is absolutely no problem to analyze this from different cases. So that you can train the model on several images from the same assay and then transfer it on further samples and see how well it behaves on these samples.
All right, thank you, Dr. Schneider. And we have another question for you. Just wondering about the overlay of IHC and IF. Sorry, we have so many questions coming in. My screen is skipping. Okay. Does that mean we can overlay, then make an app to analyze further?
Yes. So I think that you are referring to transfer learning with this question, and yes, you can do this. So you can, if this is stained on the same tissue section, like with the UltiView, or if you have a true serial cut sections, then you can co-register these and transfer the information from one slide to the next. And our co-registration is very powerful and can do this in most of the cases. And then you can learn one thing, for example, with IF to the H&E and transfer this and create your new algorithms to directly identify cells in H&E, for example.
All right, thanks again, Dr. Schneider. And there's another question, actually two questions for you, two questions in one. Is the deep learning process algorithm explainable from the regulatory point of view? And any hope timeline for FDA or CE certification of the software as a medical device for clinical diagnostics?
Okay. So I don't think that any of the deep learning process algorithms are yet explainable for regulatory point of view. We would have to look into this question and get back to you. But if you shoot me an email, I can definitely bring this to our regulatory chief of our regulatory department and discuss this with Esther Abels. And for the FDA or CE certification, we have IVD CE marked apps for IHC markers like KS67, HER2, ERPR, tumor nest detection in H&E in lymph nodes from breast cancer patients. So yes, that is possible, but you need a lot of training material and a lot of sites and need to coordinate to get this approved.
All right, thanks again, Dr. Schneider, and you'll have an opportunity to connect after this. We really appreciate you doing that. Okay, our next question is, how does the cell segmentation work? Based on nuclei expansion or also by other means?
Yes, so the current cell segmentation works on the expansion of the nuclei. So this is the first thing what we do. We can also incorporate training that you learn from the lineage markers, but this is something what you would have to do on your own currently. So the algorithm doesn't know this yet.
All right, thanks again, Dr. Schneider. Let's have another question. Can you comment on antibody stripping and reprobing and what makes UltiView so different?
Yes, so antibody stripping is something that we don't do at all. So basically we perform one single incubation of primary antibodies. So everything happens simultaneously. We do not need to do sequential staining, and therefore we do not need the antibody stripping. And this is something that can really affect the antigenicity of your tissue as well as the tissue morphology, which we try to avoid completely. So what makes UltiView so different is really the fact that we do one single step for each of the step required for the staining. So retrieval is only one, primary antibody incubation is only one, and then we also add on top of that an amplification step, which is increasing the signal to noise, and then allows a more consistent data and a better algorithm development.
Thank you, Dr. Bazzaturo. We have another question coming in. Let's see. What are the specific tissue requirements? Is the technology amenable to needle biopsy or even small metastatic tumor samples?
Well, I think this question goes very much with the previous one, in the sense that, when you are performing sequential staining and you need to repeat a kind of retrieval or use a stripping step, and you are working with very small samples or like needle biopsies, then you
are afraid that basically your tissue can fall off the slides. But in this specific case with the Ultima technology, we do not have this risk. So there is no special requirement other than the classical ones that you would have also for any other IHC staining. So you want to have slides that have been cut recently. You don't want to have epitope degradation before you perform the staining. So you need to pay attention to your slides, but also, most of it is done at the very initial phases when you collect the samples and the way you embed them, the way you preserve them, which is something typically done and taken care of for any type of staining. So the short answer is that no specific requirements are there, and we can work with very small tissues as well.
Thank you, Dr. Vasaturo. Our next question is, cells in solid tissue are usually close together or close to each other, forming clumps and or overlapping with each other. How does the Visiopharm software deal with that?
Yeah, that's a great question. So that's something that the user would then need to train also the deep learning classifier. And the more general you cut your sections at the same section thickness, the better your model will behave on your samples. But we cannot guarantee that all the cells that are overlapping with each other are identified correctly. And for this, especially when they have the same lineage markers, then it's becoming tricky for the algorithm. But for this, we would like to introduce also in the future that the users can correct for these mistakes. Thanks.
Thank you, Dr. Schneider. Our next question is, what qualifies an UltiView technology antibody versus a standard IHC antibody?
Great question. So the antibodies that we use are usually the clinical relevant clones when they are available. So basically there is no difference between an antibody that you would use for the classical IHC DAB staining or an antibody that you want to use with the ISP technology. So the proof for that is also that DAB staining is still our gold standard. So whenever we work with an antibody that is developed to work on paraffin embedded samples, then this can be developed for ISP. And we will always start from DAB, testing the sensitivity and specificity of the antibody, and then we will conjugate it with our DNA barcoding. Repeat the staining in DAB to make sure that this has not affected the performance of the antibody, and then eventually move it into fluorescence. So we can work with any antibody that has been developed for paraffin samples.
Thank you, Dr. Vasaturo. And another question, let's see, what other software can you use for image analysis of the MIS?
Maybe
I guess I would take this question. Yeah, I think I would take this question because, Visiopharm, of course, and we have seen it in action, but in principle, we are not limited to any specific software. So we work also with other partners for image analysis, and the choice really depends on the depth of the phenotyping you want to do and the complexity of the analysis you want to perform. So you might even think if you just want to look at the density of a single lineage marker, you can even use the freely downloadable softwares that are online. But of course, if you want to do a much deeper analysis and you want to understand more and you want to go into spatial distribution, proximity analysis, etc., then you need to use a software like the Visiopharm.
Thank you, Dr. Vasaturo. I'm sorry, I'm trying to read questions and talk at the same time. So the next question is, do you have any recommendations regarding section thickness to get good multiplex staining as well as cell detection?
Well, I can talk through the thickness for staining. We usually work with the classical 3 to 5 micron slide. It really depends a little bit also on the customer request. So in-house, I think usually we use 4 micron slides thickness, but there are some customers that also request 3 and some others that request 5. I don't think it changes a lot in terms of the cell detection because you are still below the size of the cell. So you know that basically if you look at serial sections, if you have 4 or 5 microns, you are not looking at the exact same cells. So maybe Fabian, if you want to comment further on the thickness and how this would impact the analysis.
Yeah, thanks. So we have tested that, and also in my previous jobs, we have tested the section thickness, and we found that the best consistency we found was 3 or 4 micron. So that was really the best, where you don't have too many cells that are overlapping or hiding each other or on top of each other. And the more you will turn into 5 and 6 micron, the more you also have section thickness artifacts, because you can't guarantee always that you cut at the same size due to temperature changes, etc.
All right, thank you both. And let's see, we've got another question. Typically, many image analysis tools are greatly influenced by inconsistent staining results. How does Visiopharm software account for inter and intra-run variability in staining quality?
Yeah, this is a great question, and we get this asked a lot. So I think our software cannot do anything about inter or intra-run variability if the variance is really high. So we, in image analysis, you rely on the input data that you get. And if these are inconsistent, then even the best models cannot take this into account. So you have some tools at hand that you can use. So for example, you can use one of our normalization tools that we recently deployed, where you can also do the normalization across images in your batch. And we are still working on how to do a good image normalization, thresholding, and have this adaptively transferred onto other images.
Well, it looks like, thank you so much. It looks like that's all we have time for today. Do either of you have any final comments for our audience?
Well, I think we have been able to convey the message that it's really important to define upfront the best biomarkers to combine, and not just think about the number of markers you can have on the same slide, but really think through the hypothesis that you want to validate, and then have a very robust and reproducible assay that eventually allows you also to have a very robust data on the image analysis side, and facilitates also the task of building an algorithm that can be applied across multiple indications and across multiple trials.
Yes, I can just echo that. And with one of the things that one of my former boss at AstraZeneca, Carl Barrett, was always saying, first think about your hypothesis before you put the panels together. And I think this is a very wise one. And first we need to all think about what is the mechanism of action or the proof of mechanism that you would like to see, because most of the phenotypes that you are hunting are already known. And if you would like to combine an image analysis with your RNA sequencing data from single cells, then you have already maybe new targets that you would like then Ultiview to test new antibodies on, or to develop new panels. And then you can either have custom-made algorithms from Visiopharm as a service, or you can develop with our modules your algorithms on your own, and then use them for further validating your new hypothesis and your mechanisms of action.
And maybe just to really close up on this, I think Ultiview has a quite strong scientific team that can help you discussing the ideal combination of markers and consult on your specific needs and really help getting the data that eventually give you the utility you need.
Well, thank you again, Dr. Vasaturo and Dr. Schneider, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Ultiview, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.